So good morning, Stefan, and over to you.
Yeah, thanks a lot, Alan. Good morning to everyone. So I'm really happy to talk about our current story about the spatiotemporal investigation of SARS-CoV-2 infection virus clearance in lungs of Golden Syrian hamster. So the main players in that work are my PhD student Ola Bagato and Dmitry Utakov, who is the head of our bioimaging lab, and who is more interested in the details of this story. It's already on bioarchives and hopefully soon in a peer-reviewed journal. So the hamster turned out to be a good model for deep lung infection by SARS-CoV-2 with all the complications we can expect after SARS-CoV-2 infection. So the bronchial epithelial cells are infected, it goes deep in the alveoli, macrophages invade, and we have destructive effects on alveolar epithelial cells type 1, and they are replaced by type 2 cells, and so on. So on. At the end, you have tissue repair, fibrosis maybe, and edema, all these complications. There's a lot of work, has been a lot of work done on that issue, and we tried a little bit different approach we wanted to follow the infection and host reactions in a more broad global way by large volume 3D imaging of the virus infection over time, and also to follow the host responses in the lungs of infected hamsters. So we did 3D imaging of large tissue volumes. We did different virological and histochemical analyses and combine it with an RNA analysis by RNA-seq to bring imaging data to molecular host reaction patterns. So what was done is a simple infection experiment in hamsters. So hamsters were infected by orotracheal inoculation, and they were monitored for till 14 days. And as we could expect it, the weight was lost until 7 days. The control animals were fine. And when we followed the SARS-CoV-2 infection in the lungs, then we saw what many people saw, that the RNA levels were quite high for a long time till day 7, but the antigen, the infectious virus levels decreased quite early, already at day 3, and decreased further with no infectious viruses in the lung at day 6. So there is a discrepancy between infectious virus release and RNA detection. So, and our pathologist, of course, looked in these lungs and also found what many people found, that at the beginning of the infection, they saw nice infection of bronchial epithelia, and later, so the virus was more found in alveolar tissues, as you can see here. So the main antigen detection in bronchia was indeed at day 1, and it was a little bit longer in these alveolar epithelial cells and decreased until day 7, but still detectable there. So these are very beautiful and great and detailed observation, but we wanted to follow the infection in the lung lobe, to see, is it everywhere the same, or can we see something different? And that's why we used our light sheet high volume immunofluorescence imaging approach. And by this technique, we can really immunofluorescence stain complete lung lobes or thick slices or even complete lungs and detect the virus in the whole tissue. And what you can see here is in green, just the background image where we see the structure, the morphology of the lung, and in magenta, the virus infection. So the virus is present in bronchi, and you have this cauliflower appearance of the virus, which reflects the infection of the alveoli. So now, having this technique at hand, we wanted to follow the infection over time. Just before I come to the kinetics of the infection, I just want to shortly introduce you in the technique. So the problem if we have thick tissues is that we cannot look through, and if we want to do imaging on it, we have to make it transparent, we have to clear it. So, and the main problem to look through something is light scattering and absorption. No? So, and that's because at every interface between different matrices, for instance, water, lipids, or proteins, the light is scattered, and if there are too many interfaces, you cannot look through something. So if you decrease this scattering, you can look through, and we do it by techniques. So we delipidate the tissues, we dehydrate, and we decolorize, remove pigments, and so on. And at the end, we have a refraction index matching. So if we remove all structure components except proteins and have a liquid medium inside that has the same refraction index than proteins, we can look through, and that's exactly what we do. So this is a pipeline we use for our studies. We have the hamsters, we take tissues, and then we make a decolorization, permeabilization. Then we stain these tissues. It takes quite a while because you have to incubate several days and wash, and we dehydrate, and then we replace the liquid solution with an organic solvent, ethyl cinnamate, that has the same refraction index than the proteins, and then we can look through, read newspapers through the organs, and then we can do really high volume light sheet imaging with a special microscope, a light sheet fluorescence microscope, which now enables us to image quite fast 3D image stacks of tissues. And after we have done this light sheet fluorescence microscopy, we can go with these samples, we can slice them and go to the confocal laser scan microscope to have higher resolution images. So that's the approach, and that's the time course of a SARS-CoV-2 infection in a Golden Syrian hamster. In magenta, you see the nucleoprotein of the SARS-CoV-2 virus. And you see exactly what pathologists saw in conventional immunohistochemistry. At day 1, mainly bronchial epithelia infected, and then at day 2, it's the cauliflower appearance of the infection, and then from day 3 on, it starts to be decreased. And we have here from the inner side of these cauliflowers loss of NP signal, and at the end, day 7, we still have antigen, but way less. And the interesting thing is that we can combine this technique with detection of host cell factors, for instance, MH2, a major histocompatibility 2, or here CD68, which is a marker for macrophages. And what we can see here is that at the beginning we have low levels of tissue-resident macrophages, but when the virus is starting to be cleared, then there we have appearance of a big wave of macrophages in the tissue. And so I come to that point later. Let's focus on day 1 here, and then on day 4. So when we look at day 1, we see that mainly the bronchi epithelia are infected, and we see a clear upregulation of MHC2 in this epithelia in infected cells. No, or almost no, in upregulation in non-infected cells. So this is connected to infection, and we could confirm this upregulation in infected cells by higher resolution confocal imaging. So that's the one thing. And if you just look here, so we see only few signals for CD68. But if we wait, we have this wave of macrophages, myeloid-derived macrophages that appear to come from the blood vessel. I come to that later or show it to you. So here again, we have still the infection, but here an airway, here a blood vessel, but the airway structure is not anymore magenta positive, so the virus already has been cleared here somehow. We still have MH2 upregulation, but here this massive wave of macrophages. So that's just an impression. So we have to take the lungs from individual image, and then we think we have a time series. But of course, at that stage, we cannot do any statistics. So we had to find a way to compare different animals for this aspect we had to quantify. And that's why we implemented machine learning algorithms in this 3D image analysis to segment these structures, to make objects out of it, to be able to quantify volumes, percentages of the tissue we analyzed. And one example that for this approach is showed here. So we have a green background image. The program learns to segment airways or blood vessels and to recognize the immunostaining signals correctly. So NP, MHC2, and myeloid-derived macrophages in that case. So, and having these approaches, bioinformatic approaches at hand, we can really digitalize our samples. And if we can do that, we can work with different animals and then quantify and compare really the extent of infection and so on and so on. And that's what we did. And here you see the time course of SARS-CoV-2 infection here in magenta in 3 different animals. And you see this nice kinetics. And what you can also see is that the monocyte-derived macrophage infiltration is linked to virus clearance somehow, because if the virus starts to decrease, the macrophages increase. And this is connected to the upregulation of specific factors that are connected to macrophages, like CD68 or Cyclic-1, for instance. Okay, then, of course, we looked also for deeper for the MHC2 upregulation in SARS-CoV-2 infected cells. And that's here we have a quantification, and we always see in the red, in the NP positive cells, also higher levels of MHC2, whereas in the negative cells, we have lower levels over the whole time. So if we look for the extent, the volume of MHC2 positive cells in non-infected cells, we have, according to the graph above, low levels of MHC2 in earlier phases, but in later phases it increased. And this indicates that somehow this is still there, although the virus is already removed. And this is, of course, connected to the upregulation of antiviral responses of the STAT-JAK pathway, which is responsible for the upregulation of MHC2. So next, we looked for also deeper for the cell types that are infected, and one cell type in the bronchi are the club cells. So back to day one after infection, where we have mainly bronchial infections. So as you can see also here in magenta, we always thought that there is something strange, because if you look here at the right side for the NP signal, there are areas in the bronchia that are not so much infected. And when we counterstain with uterogluculin, a marker for club cells, so non-ciliated cells in the bronchi, we saw that these cells were not really heavily infected. So definitely in these cases, either the club cells are not infected or the uterogluculin marker is somehow lost. I think the first thing is the case. And if we look there for MHC2 upregulation, we see again that in the infected cells in the MHC2 is upregulated, whereas in non-infected not. The uteroglobulin cells were a little bit upregulated, but not to the levels of the infected ones. And in the lower right image is a confocal image where we can clearly see the separation of staining of the virus and the club cells. So to confirm this non-infection of uteroglobulin infected cells. So, and then of course we looked deeper for the macrophage appearance. And when we segmented the blood vessels, we clearly could see some spatial relation of the blood cells and these waves of macrophages appearing to be released from the blood vessels. So, and of course, we did also specific staining of the blood vessels with against von Willebrand factors. And in the middle image, you see nicely the macrophages somehow connecting to the blood vessels. We think they are coming from the blood vessels. And in magenta, you see some minute rests of the virus antigen. And on the right side, you see a RNA-seq analysis. And at that time point, we see accumulation of coagulation factors that indicate vessel damage. So, and maybe this vessel damage enhances the infiltration of macrophages at that time point. So, and this is connected at later time points at 6 days post-infection with massive apoptosis induction, here imaged by caspase-3 detection. So in the whole lung samples, we see a massive caspase activation. And as if here we quantified this, of course, in this diagram. So there is an upregulation. And what we can also see in the RNA-seq analysis, this correlates to the upregulation of typical apoptosis factors. So there is a cell death occurring, and at the same time point, or almost the same time, and that's, I think, important, we have also cell proliferation going on increase. So at 7 days post-infections, we see by imaging the Ki-67, a marker for cell proliferation, massive cell proliferation, not at the sites of the infection, not at the sites of the macrophage, but everywhere around. So maybe at sites where the virus already has been removed. So, and this is also correlating with increasing upregulation of this Ki-67 protein marker as indicated by RNA-seq analysis. And this also correlates with histopathological observation that at 7 days post-infections, we have still a clear pathological appearance of the lung tissues with massive regeneration and apoptosis. And at the end, in blue, you can see the collagen staining, which ends up in fibrosis after 14 days. So I'm already at the end. So what I wanted to show you today is our approach to make a spatiotemporal analysis of the dynamics of SARS-CoV-2 infection and host response. So we observed a wave of myeloid derived macrophages that appear to be linked to virus clearance in connection with tissue injury and blood vessel damage. Rapid initiation of prodromic factors, upregulation, tissue repair, and alveolar proliferation results in tissue remodeling, followed by fibrosis. And we think, although the hamster resolve SARS-CoV-2 following the MDM influx and repair lung tissue integrity, longer-term alterations of the lung tissues may arise as a result of concurrent tissue damage and regeneration process. And lots of people were involved. So at FLI in Greifswald, but also RNA-seq was done in the Paul-Ehrlich Institute in Langen and also the University in Bochum was involved, and also people of in Greifswald University supported this study. I thank you for your attention, and I'm happy to hear questions.